Biotechnology, and I'll be moderating today's session. We appreciate the diverse participation of our audience today who have joined us from across the country. Please feel free to share your questions and comments using the chat feature. We will address as many questions as possible during the question and answer session. For today's webinar, our focus is on biomanufacturing of enzymes, both academia and the industry perspective. We are delighted to have an esteemed panel of speakers with us who will share their expertise. From DBT, we have Dr. Alka Sharma, Senior Advisor, and Dr. Amit Yadav, Scientist D. From academia, we have Dr. Shams Yazdani, ICG. And the industry perspective will be shared by Mr. G.S. Krishnan, President of Association of Biotechnology-Led Enterprises, ABLE. So I now invite Dr. Alka Sharma, Senior Advisor, DBT, to set the context for today's session. Over to you, ma'am.
Thanks, Vaishali. Namaskar. Good afternoon to all of you. On behalf of Department of Biotechnology and BioRec, I welcome you all for today's webinar on biomanufacturing of enzymes under the webinar series on Bio-foundry and Biomanufacturing initiative of Department of Biotechnology. Many of you may be aware the Union Cabinet under the chairmanship of our Honorable Prime Minister approved India's first biotechnology policy, that is Bio-E3, Biotechnology for Economy, Environment and Employment policy for fostering high-performance biomanufacturing in August this year. The Bio-E3 policy was released by the Honorable Minister of State, our Honorable Minister of Science and Technology, Dr. Jitendra, August 2024, this year only. So subsequently, the Union Cabinet has also approved the umbrella scheme of the department, that is Biotechnology Research, Innovation and Entrepreneurship Development. We call this BioRide in September this year. Biomanufacturing and Bio-foundry initiative is one of the new components under the BioRide scheme with a budgetary outlay of rupees 1,500 crores for 3 years, that is up to March 2026. This policy will set the Bharat at the forefront of future that is more sustainable and responsive to global challenges by accelerating and harnessing biomanufacturing solutions that encompass diverse bioeconomic activities while safeguarding environmental and climate impacts. This Bio-E3 policy aims to set forth a framework that ensures the adoption of cutting-edge advanced technologies and to align innovative research and aim at revolutionizing the biomanufacturing processes for enhanced efficiency, sustainability, and quality, while also accelerating development and production of bio-based high-value products. The timely policy will position India as a global biomanufacturing hub, steer India on the path of accelerated green growth, fast-track innovation to technology sustainability, drive employment and intensify entrepreneurial momentum, achieve bioeconomy targets and national economic goals for 2027, and create a bio-vision for Bharat. The policy aligns with the Honorable Prime Minister's clarion call for lifestyle for the environment and the Union budget emphasis on green growth. The interim Union budget for 2024 and 25 financial year has also announced Bio-foundry and biomanufacturing as a new scheme. Through extensive national consultation and sectoral expert meeting, 6 thematic sectors have been identified for biomanufacturing initiative. They are bio-based chemicals and enzymes, smart proteins and functional foods, precision biotherapeutics, which includes cell and gene therapy, monoclonal antibody and mRNA therapeutics, climate resilient agriculture, carbon capture and its utilization, and futuristic marine and space research. A strategic roadmap has also been formulated to implement these thematic verticals under the Biomanufacturing Initiative. Call for inviting proposals under these thematic verticals will be announced as soon. The first BIREC DBT Joint Regulatory Conclave was also organized for the smart protein in April this year. The similar kind of regulatory conclave we are planning to organize for each thematic sectors and subsectors. The policy also envisages setting up a bio-enablers such as bio-artificial intelligence hub, bio-foundries, and biomanufacturing hubs to support innovation and scale up across the thematic verticals. The Bio-Artificial Intelligence Hub will focus on powering data-driven research across the thematic verticals, while biofoundries will be set up for early scaling up of proof of concepts, and biomanufacturing hubs will be established as a shared pilot scale and pre-commercial scale facilities to support scale up of bio-based products in the identified thematic sectors. I'm happy to share that the first DBT BIREC joint call for proposal entitled Mulankur Bioenablers, Biofoundries and Biomanufacturing Hub was issued, and as an outcome, large number of proposals have been received so far, which are under evaluation jointly by DBT and BIREC. India's first biomanufacturing institute, that is the National Agri-Food and Biomanufacturing Institute in Mohali, was inaugurated and dedicated to the nation this year in October by our Honorable Minister of Science and Technology, Dr. Jitendra Singh Ji. And a memorandum of understanding has also been signed between Department of Biotechnology and ISRO and Department of Space for cooperation in space, biotechnology and biomanufacturing in October this year. And DBT is in the discussion with the other ministries and departments for establishing the in the identified thematic areas. Outreach activities for Bio-3 policy are being carried out extensively through press media, social media platforms, editorials, school campaigns, and all to highlight its significance. Vartalap has been organized at few places in the country to interact with the stakeholders and highlighting the importance of this Bio-3 policy as a futuristic approach. To broadcast the activities under Biofoundry and Biomanufacturing Initiative, a website has also been launched. You may visit the website through DBT website, and there's an icon for biomanufacturing. To further disseminate information about Bio-3 policy and discuss the thematic verticals, DBT and BIREC are jointly organizing the webinar series on Biofoundry and Biomanufacturing Initiative. This series features discussions by DBT, BIREC officials, and scientists on thematic sectors and subsectors, emphasizing their potential impact on India's bioeconomy. It provides a valuable platform for researchers, startups, SMEs, and industry representatives. Today's webinar, the fourth webinar in the series, is focusing on biomanufacturing of enzymes. Biomanufacturing of enzymes is a sustainable and efficient process that uses biological systems such as microorganisms to produce enzymes for industrial applications. For the past 2 decades, there has been a growing interest in replacing chemical catalysts used in several industrial processes with more efficient and environmental friendly alternatives. As industries increasingly focus on sustainable practices, biomanufacturing of enzymes plays a critical role in reducing environmental impact while supporting innovation across multiple sectors. Once again, I welcome each one of you for today's webinar and hope you will find this discussion insightful and inspiring. Thank you very much.
Thank you, ma'am, for providing an excellent overview of the biomanufacturing initiative. Our next speaker is Dr. Amit Yadav, Scientist D at DBT, who will discuss about the enzyme subsector of biomanufacturing. Over to you, Dr. Yadav.
Thank you, ma'am. Good afternoon and welcome to the fourth webinar under the webinar series on biomanufacturing and biofoundry and the biomanufacturing of enzymes. Next slide. When we talk of biomanufacturing, first question that comes to mind is why there is a need to transition from traditional manufacturing to biomanufacturing. And this will be clear from this slide. Traditional manufacturing relies on non-renewable fossils, leading to rise in greenhouse gases, while biomanufacturing utilizes renewable sources. Traditional manufacturing requires mega facilities operating at high temperature and pressure, in contrast to biomanufacturing, which require fermenters operating at low temperature and pressure. Traditional manufacturing runs on high capital expenditure, while biomanufacturing requires low capex. Traditional manufacturing is not amenable to sustainable innovation, while biomanufacturing leads to sustainable innovations. Traditional manufacturing is localized in nature, while biomanufacturing facilities can be distributed in Tier 2 and Tier 3 cities. So it can be said that transitioning from traditional manufacturing to biomanufacturing presents an opportunity to create a more sustainable industrial landscape. By embracing renewable sources and innovative practices, we can mitigate environmental impacts while fostering economic growth in diverse sectors with cleaner, greener, and sustainable solutions. Next slide. When we talk of enzyme biomanufacturing, it offers many advantages over the traditional chemical processes. Enzymatic solutions are sustainable in nature. Enzymes typically operate under milder conditions of temperature and pressure, which minimizes energy consumption and reduces the environmental impact associated with traditional chemical processes. Additionally, enzyme catalyzed reactions produce fewer toxic byproducts, contributing to lower ecological footprint. Enzymes have high stereoselectivity, which yields stereo and regiochemically defined reaction products. With the advancement in genetic engineering, development of enzymes with improved stability and activity under various conditions is now. This includes enhancing thermal stability and optimizing pH levels for specific chemical applications, which is crucial for industrial processes which involve extreme environments. Enzymes are also used in many industries. They are employed in a wide range of sectors, spanning from food, pharmaceuticals, textiles, to biofuels, to name a few. In paper and pulp industry, enzymes like cellulase, xylanase are used for improved bleaching and improvement in fiber properties. In detergent, protease, lipase, amylase are there to remove stains. In pharmaceutical, enzymes like nitrile hydrolase, ketoreductase, peroxygenase are increasingly being used to catalyze specific chemical reactions, which are right now not sustainable. In food processing, trypsin, amylase, pectinase are increasingly being used to convert starch to glucose, high fructose corn syrups, deep-bittering fruit juices. In biofuel, cellulase converting cellulose to glucose is ultimately converted to ethanol for its subsequent uses. When we look at the global and the Indian perspective, India's enzyme bioeconomy is approximately 3 to 4% of the global market size. However, the Indian market is poised for growth through increased production capabilities and expanding applications across multiple industries. According to the Indian Bioeconomy Report 2024, significant growth has been seen in various sectors, particularly the textile and food industries, which leverage enzymatic processes. The textile sector grew by 18% to achieve a market valuation of 8.2 billion. In the food sector, the bread and the biscuit segment grew and reached 2.68 to 0.8 billion respectively. Next slide. Now, the major gap areas that need to be addressed in enzyme biomanufacturing, the first and the most important is the high cost of production, which is mainly attributed to expensive substrates which are used in enzyme production, low yields of enzyme, which are which we got after the fermentation, which basically requires larger volumes of raw material and thereby increase the cost of enzymes. However, achieving high titers is essential to make the process economically viable. Subsequently, in downstream processes also, the purification, formulation, ensuring adherence to quality standards and regulatory requirement increases the cost. This high cost also then leads to the dependence on imports. Most commercially available enzymes are derived from mesophilic organisms, and they are stable only within a limited range of conditions. So they tend to lose activity quickly under extreme temperature, pH levels, solvent concentrations, which we encounter during industrial applications. So maintaining efficiency, stability is a major issue which needs to be addressed. Many enzymes also require cofactors to function effectively, and these need to be replenished continuously during reaction. This again adds additional cost and complexity to the production process of enzyme. Now, in the scale-up process, transitioning from lab scale to industry scale pose significant challenges. Maintaining consistent growth condition, enzyme activity at larger scale can be difficult, often leading to variation in product yield and quality. The robustness of microbial strains used in fermentation is critical, as any instability can result in product loss. In the regulatory issues, the enzyme biomanufacturing critical to ensure safety, efficacy, and compliance with country-specific regulations. So these were the major gap areas that need to be addressed. Next. To address some of the gap areas, newer technologies and methods have accelerated the future commercial production of enzymes. Now, novel enzymes are accessible through numerous microbes, inhabit the biosphere. Only few are cultivable through standard lab techniques. However, now, because of metagenomic approaches, has led to an alternative strategy to conventional microbe screening by preparing genomic library from environmental DNA and systematically screening such libraries for open reading frame, potentially encoding putative novel enzyme. Genomic sequencing program have also resulted in explosion of information available from sequence databases, thus creating opportunities to explore or possibly finding newer enzymes by database mining. Extremophiles are a very interesting source of enzymes with extreme stability under conditions regarded as incompatible with biological material. Now, methods for production of cost-effective synthetic genes are also available for rapid, affordable screening of enzyme variant using high throughput screening tools. Targeted protein engineering involving site-directed mutagenesis and directed evolution are also there. Now, AI is increasingly playing an important role in the prediction of enzyme structure and its corresponding characteristic and functions, which will make the design of artificial enzymes more accurate. Next slide. Next. Thank you.
Subsector. It is now my pleasure to welcome Dr. Shams Yazani, ICGB New Delhi. Dr. Shams is a distinguished scientist and group leader at ICGB New Delhi. His research primarily targets the development of microbial strains for producing biofuels, biochemicals, and other value-based products from renewable resources. Over to you, Dr. Shams.
Thank you very much, Dr. Vaishali. Is my slide visible?
Yes, sir.
Okay. So, first of all, thank you for giving me this opportunity to present in this very, very important webinar. I'm going to basically focus on the academic perspectives and majorly trying to give you an importance of microbial strain and protein engineering in the enzyme manufacturing. Already Dr. Amit has very nicely showed the importance of enzymes in different sectors, and we don't realize that every day where all enzymes have actually invaded in our life, including from the households to the clothes that we wear and the biofuels now that we are using in the pharmaceutical industries, animal feeds, everywhere, and especially in the food and beverages, it has really been started being used in a very large quantity. So therefore, it is very, very important actually to understand how these enzymes can be made in a much, much larger way at a much, much cheaper way. Now, just to give you some sense on where the enzyme market globally and Indian perspectives they stand, and of course, it is growing at a very large rate of CAGR of 6 to 7%. At this moment, it is $15 to $16 billion US to billion-dollar market. And almost half of the global enzyme market is dominated by Novozymes, and the other half is by some other enzyme companies. And in India also, this enzyme market is increasing slowly. It is increasing at a rate much higher than the average global rate at 10% CAGR. What is most interesting and important for us to understand is that India is also the topmost importer of enzymes. So this data of ships arriving in India, which I got it from Volza, it says that India is the topmost importer of enzymes throughout the world. And around more than 80% of shipments are imported from China. So of course, there is a need to understand that why we are discussing about biomanufacturing of enzymes in India. Now, if you look at what is the platform by which enzymes are produced globally, so almost 85% of enzymes are coming from different microbes, microbial sources or microbial platform, be it bacteria or fungi or yeast. And then the other, 15% are produced via plant and animal. So this also highlights that microbial platform play very, very important role in the enzyme production. So given that there is a need to enhance the capacity and innovation for biomanufacturing enzymes in India, what should we do to enhance those? So I have listed some of those pointers. For example, we certainly need to develop engineered host platforms that are non-pathogenic in nature and non-toxic in nature. Normally, most of these enzymes are produced in a very large quantity in a bulk production fermenter. Being a non-pathogenic, non-toxic in nature is very, very important. Also, if you want to engineer the host platform for higher enzyme production, we need to have tools. Most of the tools that are available, they are all highly patented, and therefore there is a need to develop new and novel tools for efficiently and reliably engineer the host platform. And also we need to have expertise to do synthetic biology so that we can increase the kinetics and the thermotolerant properties of all those enzymes which Dr. Amit nicely mentioned in his presentation, like carbohydrases, proteases, lipases, polymerases, nucleases, etc. And the most important thing, especially for the bulk production and very large fermenter operation, is that we need to develop antibiotic marker-free organisms so that our environment is safe. The antibiotic markers are not really transferred from our host organism to any other wild organism. Now, the last but not the least in this list is promoting the idea of what I call it as a design, build, test, and deploy scheme. Now, here, there's basically a hybrid of what Dr. Alka said about Bio-foundry and biomanufacturing. So Bio-foundry, where we work on design, build, test, and learn cycle to keep improving our host organism. And then biomanufacturing basically is leading to deployment. So we need to collaborate. We need to shake hand between the team that is working on Bio-foundry and the team that is working on biomanufacturing so that we can eventually have a design, build, test, deployment scheme to facilitate faster market entry. Now, with this, I'm going to give my own journey as an example of developing enzymes that, it was almost a decade of work. We work on enzymes that are responsible for hydrolyzing lignocellulosic biomass, so basically all the agricultural residues like rice straw, wheat straw, corn stalk, or corn cobs, or sugarcane bagasse. They're all made up of lignocellulosic biomass. And we know that in India, they are produced really, really in surplus. as much as that we burn and we actually release a lot of carbon soot in the environment, leading to global warming, and therefore finding a solution for this is very, very important. If we use the lignocellulosic biomass, especially the carbohydrate portion of that is present in the form of cellulose and hemicellulose, if we can convert that into the monomeric sugar, then we can ferment it into useful product like ethanol or any other thing. So enzyme is very, very crucial to convert these polymers into monomers. Now, of course, lignin is something that we need to take care of. There are enzymes that degrade lignin, but they are very, very poor in their catalysis. Therefore, this step is still done through thermochemical treatment. But cellulose and hemicellulose, now there is a huge research that is going on to produce enzymes that can do this work of hydrolysis much faster. The issue is that there are not one or two enzymes, there are a series of enzymes that are involved in hydrolyzing the cellulose and hemicellulose into monomeric sugars. Some of them I have listed here, like cellobiohydrolase, glucanase, beta-glucosidase, LPMOs, and all that, and similar to xylem. Now, the issue is if there are one or two enzymes you can heterologously express in some very, very industry-friendly host. But if there are multiple enzymes, dozens of enzymes, then you really need to find solution in the nature and try to screen the organism that will produce most of these enzymes. And that's what we had done with the help of DBT. We had a bioenergy center, which had a high-end equipment like robotic colony picker and robotic liquid handling system, which we use to screen large number of natural isolates. India is full of biodiversity. So we isolated and tested number of bacterial and fungal isolates. We used some mathematical model to make sure that the organism that we are testing is producing enzymes, known enzyme like endoglucanase, exoglucanase, beta-glucosidase, and also because they are the not the only enzyme, there are many auxiliary enzymes. We also wanted to test the biomass hydrolysis potential of those enzymes by looking at the sugar released by the biomass hydrolysis. And through all that testing, we tested the weighted some matrix score for each of these isolates, and we picked up one of them, which was earlier found to be Penicillium phenoplosum, but after thorough genome analysis, we realized that Spinophilus species. Among all the species that was outstanding, and therefore we went ahead and did the genome sequencing, annotation, and very, very deep proteomic analysis to understand what are the kind of enzymes that it produces. We purified many of these enzymes and characterized and compared with its peer enzymes, and we realized that this host is really, really nice and produces enzymes that are superior to many other enzymes. And therefore, we went ahead and started developing tools to engineer the genome so that we can increase the efficiency of enzyme production. And of course, being a wild-type organism, it was extremely difficult to engineer the genome just because it was tolerant to all the antibiotics we normally use in our transformation system. And therefore, domesticating this particular species took some time, but eventually we were successful in developing tools, various tools to engineer the genome, including the popular CRISPR-Cas, and there were several other tools that were also discovered. And eventually, we were able to engineer the genome in terms of deleting the repressor transcription factor that was responsible for repressing enzyme production, as well as engineering some of the key enzymes that it was producing and putting that back into the genome, like cellobiohydrolase, lysate polysaccharide monooxygenase, the engineered beta-glucosidase, making it much more thermostable. And therefore, over the period of time, we were able to improve the secretion ability of this particular host to a very good enzyme titers. And not only that, most of the time we fail in the scaling up of the process. We showed that our technology that we developed is highly scalable. Without losing the expression ability, we were able to scale it up to 15,000 liters scale and tested our enzymes to a various biomass, including the biomass that was coming from one of the first refinery, the second generation ethanol commercial plant that is made in the Panipat refinery. We tested and compared with the best commercial enzyme that was available, Our enzymes, the engineered fungal enzymes, are as good as the commercial enzymes. And after addition of engineered thermostable beta glucosidase, it was further improving the biomass hydrolysis. So, of course, in this journey, Praj was helping us in the validation, and we are talking to HPCL and BPCL how we can take this further into usage in the second generation biofuel commercial plant. So, I would like to acknowledge my team, my lab people who are responsible for what I presented in the last 1 decade of this work. And I'm also very, very thankful to collaborators and the funders, especially Department of Biotechnology, that was extremely helpful in supporting us and believing in us. So thank you very much.
For providing an excellent overview of the enzyme subsector from an academic perspective. I would now like to invite Dr. G.S. Krishnan, President, able to present the industry perspective. With over 3 decades of experience in bioindustrial sector, he has held significant positions, including regional president and managing director at Novozymes South Asia. He is a passionate advocate of sustainable biotech solutions, focusing on addressing climate change, food security, renewable energy, water conservation, and greenhouse gas reduction. With this, I invite Mr. Krishnan.
Very good afternoon, and thank you all, DBT and BIRAC, for putting together such a wonderful and very relevant program, and really appreciate the time given for me. In fact, first of all, before even starting, I would like to acknowledge and compliment both Dr. Amit and Dr. Sham's presentation. I think that gives a better perspective on the overall challenges from an enzyme perspective, and then how long it takes for innovation. And as Dr. Sham was saying, he was working on this for a decade. So it is important for everybody to understand that enzyme manufacture is not something which one could look at it at a 2 or 3 years sort of discovery. It takes a long time. And of course, the next stage in the case, for example, of Mr. Shyam's discovery on the cellulase enzymes will be the scale up which they have already achieved, and now to bring it to the commercial reality. So it is a big process which we need to understand. I will not talk about much on the technical aspect, which have been very clearly communicated. So I will cover the topics predominantly on enzymes and penetration in the Indian market. And then, of course, I will also talk about understanding the industrial enzyme manufacture, which some of it has already been shared, the source and the supply chain part of it, the role of local innovation and opportunities, and the long term strategy and Bio E3 policy and biomanufacturing. So, this is what I would be covering. So, as already mentioned very, very clearly from both the presentations, earlier presentations on the various segments of enzyme application, starting from household care, the textile, the food, especially baking and starch, beverages, beer, alcohol, wine and juice, and of course, animal nutrition, leather, ethanol, and pulp and paper. In fact, we always tell a story that enzyme is part of your day to day life, right from the morning breakfast cereal, which most of us have bread, which has actually enzymes playing a critical role, to the juice which you consume today. Actually, pectinase enzymes plays an important role on the extraction, followed by the breakfast. You go and wash your clothes, and then enzymes play an important role in removing the harsh sort of stains and others, and biotechnology and enzyme play an important role over there. And then, of course, followed by that, the jeans which you wear, or the knit segment and knit garment which you wear, each of this enzyme plays a very, very critical role. And we transformed the so-called stone wash to enzymatic wash 2 decades before, and that's again something which is a very important discovery at that time. Followed by that, again, I would say the animal feed, especially the phytase enzymes in use for the poultry industry, This is something I would like to share with you all, a very, very interesting observation. Elsewhere in the world, the phosphorus emission in the environment was a big regulation, and they had a control. And so they had to come out with some enzyme which will probably take care of fighting the phytic phosphorus. And this is where one among the few companies, including Novonesis, came out with the enzyme phytase. And globally, it was a very big hit, and the industry was very much looking forward to sort out their challenges from an environment perspective. And when we launched here in India, the first question which most of the farmers or the poultry farmers were asking us was a simple question: who's concerned about the environment with this little of bird shit? That's how it started. And then we had to tell them that it is going to be a serious sort of issue which one needs to consider in the long term. At that time, the acceptance level was very, very less. And then, you know how we got into selling these enzymes? Suddenly, after 2, 3 years, we had the mad cow disease which broke. And then the dicalcium phosphate, which was the phosphorus source for the poultry segment, they stopped using. They cannot use it. It was not available. It was a byproduct of gelatin manufacture. And then we realized this is the opportunity, and all the industry people came to us, and that's how phytase in India grew. Why I'm saying this? At the beginning, I would say almost 2.5, 3 decades before, the penetration for the biological solution was a very big challenge. The mindset to change was a very big issue. With all this, we came over to ensure every segment of the industrial enzyme application was taken care of, and we were able to at least make sure that the penetration in the market was far more. Well, no doubt, actually, China was far ahead of us, but we managed to ensure that. Sorry, I think my screen, something went wrong suddenly.
It's on, sir,
Hope
now.
you can all see my screen now. Yeah.
Yes,
Yeah,
yes,
we
yes.
can see.
Yeah, sorry. So that background why I'm giving is that's how it started. And then I should say that Novonesis, the company which Sham had already mentioned as leaders in this segment, in fact, way back almost 15 years before, the Department of Energy in US decided that they have to look at alternative sources of fuel, and ethanol was considered. That's where they came to some of the leading enzyme manufacturers to say that your cellulose enzymes cannot be viable. You have to bring it 1/10 of the cost. That's where again research, innovation, optimization, all this went into play to make sure that we brought down the cost by almost 1/10 after about 3, 4 years of research. So it is important to understand that biotechnology plays a very, very critical role, and the process it goes through to succeed, it takes quite a bit of a time. Coming to the Indian enzyme market, 75%, almost 80% today is imported enzymes. And of course, the reason behind that is, of course, some of the local companies, of course, are doing a very good job, but then they are specific to certain markets or certain segments of application. The remaining are being imported. And the leading enzyme manufacturers are covering almost all segments of the industrial enzyme application. So, this is critical to understand as far as the Indian market is concerned. The penetration or application of enzymes in almost every segment which I have mentioned here are all using enzymes. And that is far ahead of what we saw about 20, 25 years before. Also, we need to understand, I'm talking about almost 10 years before. For every dollar of enzymes we brought into the Indian market, we had to pay the government of India a duty of $1.8, which means enzyme which would cost $1 elsewhere in the world was almost $3 in India. And then, of course, with the WTO and the government's initiative, brought down the enzyme duty, which has now come down. And today it is actually viable for every segment to use. And I would say that critical segments mentioned over here, except, of course, the detergent, there are still non enzymatic detergents, whereas in the case of baking or the starch industries or the beverage, all of them are using enzymes in some way or the other applications in the various areas of the process. So, understanding the enzyme manufacture, of course, very well. articulated by both Dr. Amit and Dr. Sham about this. Of course, predominantly it is Europe, and of course, Denmark plays a very, very critical role. And then, of course, the Chinese market, which is, of course, always from a stability point of view or from a quality point of view, has always been a challenge. But otherwise, from US and Japan, there are always products coming in. So these are again very, very select countries which are actually manufacturing enzymes in a big way. And of course, India, we do have a few companies doing it, and most of them are choosing to formulation, getting the concentrate and doing local formulation. So the major enzyme manufacturing, as probably both my previous presenters will agree, is submerged fermentation. In fact, the solid state fermentation is almost disappearing. It's more submerged fermentation, again from the point of view of the quality of the enzyme, the stability, and Dr. Amit was mentioning about making sure it is thermostable and the pH control. So this is one sort of fermentation which basically requires all attention and something which has to be more precision fermentation, and that's what is happening. But the critical aspect here is to set up a facility of a submerged fermentation with a good volume of enzymes across segments. Definitely, it will be more than 1,000 crore rupees to set up a facility. And we have also done enough of survey to understand the total enzymes of various applications put together still cannot take care of the complete sort of uh, enzyme capacity if you set up a facility like this, which means only companies which have the capability to withstand or they have the opportunity to export. This is where actually setting up a full-fledged enzyme is going to make a big difference. And then I should also tell here most of the multinational companies have a good presence over here, and some of them have already are looking at submerged fermentation, which of course will address our requirement far more, and probably the cost of using enzymes, especially in the biofuel, should probably come down. And of course, we also have a few local manufacturing facility, which I've already mentioned. The role of local innovation and opportunities. In my mind, I think innovation should be on niche areas. Specifically, of course, biofuel is one among the areas which required, and what actually Dr. Sham and his team had done is definitely remarkable and something which is very much required if the government's vision is to make sure that we achieve 30% ethanol addition in fuel by 2030. It definitely requires a lot of local innovation, and that's something outstanding. However, I would always say there was always a question that do we need to again look at all replacing all those enzymes? Let me underline and tell this is not an easy task, and for local innovation on already invented sort of enzymes and its application, I would say it's only a waste of time. On the contrary, they should look at application development specifically addressing the Indian challenges and should be a focus area for us here in India. Biocatalysis is an area of opportunity, especially in the pharma segment and then the specialty chemicals area. This is one area today, predominantly the products are being imported from China, and there have been always challenges from a cost perspective or a quality perspective or a stability perspective. Some of the local companies are producing small quantity enzyme, but then they are looking at more their in-house consumption. And as already mentioned, and even by Dr. Amit, lipases, peroxygenases, and the other enzymes play an important role. These are low value but high value enzymes, which has a very big opportunity not only to look at our local needs, but also look at export market, which probably will be far more viable to produce here locally. And today, as I mentioned, majority of the enzymes requirement comes from China. So the long-term strategy and the Bio E3 policy and biomanufacturing, as already Dr. Alkaji has presented, this is one of the biggest, I would say, initiative by DBT along with the Government of India and Science and Technology Ministry to announce this Bio E3 policy. This is definitely going to encourage in a very big way some of the startups who don't have an opportunity to scale up, and some of them who basically would like to collaborate. And this is an opportunity which I think is very much beneficiary and required for the industry. So we need to encourage industry academia collaboration in this segment more precisely. And I think what actually Dr. Sham had presented is a classic example of support from DBT and also from the other corporates, which is what is going to make it more successful. Local companies should collaborate and work with MST industry leaders because there are certain innovative ideas and local scientific capabilities which probably to implement and take it to the commercial level would be a challenge. This is where collaboration with industry leaders will make a very big difference, which I think DBT should facilitate. Focus on application development, as I already mentioned, without actually, because to look at specifically local challenges today, if you look at municipal solid waste or bio CNG, these are some of the areas where one could look at it as an opportunity, and which I think a local application development with using the existing enzymes will make a big difference. And also, as I was mentioning, some of the MNCs are looking at setting up their manufacturing facilities over here. This will rather encourage MNCs to set up local manufacturing with incentives. And I should say that we have at least very clearly realized India is still a cost effective alternative. In fact, I was part of one of the companies which were actually approached for the alternative protein manufacturing locally. Still, people realize that India is a cost effective alternative with also capabilities with quality and other parameters very, very high. This is an opportunity which we should probably encourage, which will, I mean, at least see that India is the biomanufacturing hub for all the biotech products going forward. No doubt it is definitely a challenge. One should look at it, but this is where we need to work together and focus on areas which is a low hanging fruit. And of course, alternative protein is a segment which I think India could play a very, very critical role going forward. With this, I would like to stop my presentation. And precisely this is what I would like to say from an industry perspective. And I see an opportunity for niche areas for India and also to do application development. And with this, I would like to stop, Dr. Vaishali.
We will now open floor for questions from our audience. Let's take the first question. Over to Bharat.
So the first question is from the Dr. Alka ma'am. This is from the Dr. Bhavisha. How can the Bio E3 policy help startups which are working in the biomanufacturing of enzymes?
Thank you. Thank you. This is very, very important question because the overall policy is, if you see Bio E3, biotechnology for economy, environment, and employment. So this bio startup, as far as a startup is concerned, so we are now the setting up a Bio-foundry and biomanufacturing hub is one of the very important component of this policy. And under biomanufacturing hub and also Bio-foundry, we have now a special component for supporting a startup in the sense that they can come with their proof of concept or any research lead or any kind of that they think that will is going to work and work in the Bio-foundry, and then the support will be provided to the institution to help and to facilitate the activities of a startup who all are coming to use the Bio-foundry and biomanufacturing hubs. Apart from that, there is also in the thematic areas, the under the policy, as you have seen, there are 6 thematic areas, and also there are sub thematic areas. For example, bio-based products is one thematic area under which we have specialty chemical, enzymes, APIs, and biopolymers. Similarly, functional food, smart proteins, precision biotherapeutic, cell and gene therapy, monoclonal antibodies, mRNA therapeutics, and climate resilient agriculture, carbon capture utilization, marine and space research, as I mentioned in my remarks initially. Under these subsectors also, there is a provision that we are going to place the advertisement very soon. So in the advertisement, there will be provision to support a startup up to 50 lakhs as grant in aid if they want to come up with the proposal under any of the subsectors and the main sectors of Bio-3 policy. So 50 lakhs is grant in aid. Beyond 50 lakhs, if the startups, SMEs or industry, they want to do some project, they have to contribute 30% upfront in cash. So for a startup, the question was for a startup. So it's a good opportunity for them to come forward and just see where they, under which thematic areas and sub areas their proposal will fall in, and then how they can leverage this opportunity and get this kind of grant in aid from the government. Though yes, and needless to say that this will be based on the due diligence processes, and also we get comments from the experts, so that the process, stringent process is in place both in DBT and BIREC. But if there is a merit in the proposal, no one is going to deny it to that, and then they will be eligible to get the funding. Thank you. Over.
Thank you, ma'am. So the next question is for Dr. Yazdani. It is from Dr. Himanshu Goyal. He is asking, biological processes are more prone to contamination. How can we overcome this?
Yeah, of course, biological processes are prone to contamination. So you have to be very, very sure about your quality control measures. So right from making the cell banks, or every step, you have to ensure that you're not introducing any contamination. And your organism should be robust enough. For example, the fungal isolates that we work on, we realize that, if you are growing them in the right environment, there's a very little chance of other contaminant to outcompete this organism. So both the things, one is that you should have very good quality control measures throughout your process to ensure that there is no contamination happening. And second is your organism should be robust enough to outcompete any contaminants that by chance fall.
Thank you, Dr. Syed. The next question is for Mr. Krishnan by Dr. Praveen Kumar. His question is, the cost is a major factor for import substitution of enzymes. How can the Indian startups and industries make the enzymes biomanufactured in the country affordable?
Well, I don't know, first of all, which particular application you are looking at. However, what you said is right, where we are paying today about 15-20% duty come GST put together. However, I can only say some of the enzyme companies, as I already mentioned to you, are going to do local manufacturing in the next 1 or 2 years, which probably will answer your question. However, Alka ji has given a very good thought. If you have a sort of innovation and thought, and we can probably find out a way to look of scaling up and local manufacture. But I should admit, it is not going to be so easy with a particular enzymes for a particular application to go ahead and set up a commercial facility. So I would only say that you have to wait for any local manufacturer which is happening, or any other organization, local manufacturers in the enzyme segment who could probably support you in this.
Thank you, sir. The next question is for Dr. Shams Yazdani again. It is from Dr. Arushi. She is asking, how can directed evolution increase the efficiency of enzyme?
Yeah, you have to look at the kind of platform that is used for directed evolution. So, for example, if you're looking for a particular protein, improvement in the quality and the efficiency and the kinetics of one particular enzyme or protein, so you actually put that in a suitable platform and allow it to go through the directed evolution. So the way we do it, we put it in a right environment. For example, if you want this to work at a higher temperature, you should put it in an organism that can grow at a higher temperature, and then you allow it to operate at a longer period of time and allow it to evolve naturally over the period of time. And then you will find that over the period of time, your enzymes would be much more suitable for example, to work at the higher temperature. So normally the directed evolution is used for strain. For enzymes, normally people either use rational engineering, you do in silico design and rational engineering, and also you have error prone PCRs and all that. So there are different ways you can design. ALE is one, other thing is rational engineering, and third thing is the error prone PCR. Ideally, you should have a very good assay system. The most important thing is you should have a very good assay system to detect and finalize the quantity and the quality that you want in your enzymes. Thank you.
Thank you, sir. The next question is for Dr. Alka ma'am. Question is from Dr. Vipin. Is asking, how will this Bio-E3 policy facilitate the regulatory approvals for enzymes for different applications?
So as I mentioned, the regulatory reforms, the case-to-case basis is very, very important in this. And I mentioned that we organize regulatory conclave for smart proteins, where we invited the officials, senior officials from FSSAI. So DBT is working with all ministries and departments and also with the regulators to understand the issues from the applicants, and the roadmap has been prepared just to identify the gaps and challenges, and where regulatory reforms was one of the main component, that just to understand what exactly has to be discussed with the regulators to streamline the processes. So definitely DBT is going to work with the regulators in each of the subsectors and sectors, and try to resolve just to bring the invention and also the innovation to the next level, because the bridging the gap, that is scaling up, is one of the major component of this Bio-3 policy. And we all understand that for that, and the next is pre-commercial and then finally commercial. Unless we have regulatory reforms sector-wise, especially and also including this enzyme sector, we will not be able to achieve the target which we are planning to do that. Very important question, but we are working from DBT and BIREC and with the regulators in the country, and specific that gaps and challenges are being addressed. And we are also planning to have regulatory conclave in each of the sectors with the relevant stakeholders. Thank you.
Thank you, ma'am. The next question is for Mr. Krishnan again. Dr. Chandel is asking, how can Indian companies compete with global players, mainly MNCs, in the enzyme manufacturing sector?
See, what is important, as I was mentioning in my presentation, just discovery of one enzyme and setting up a facility is not going to be cost viable. It is important that you have solutions across industrial segments. And then, of course, you need all the support to set up such a commercial facility. Unless you have the capability to withstand the pressure on the volumes and everything, very difficult. Today, most of the companies, they are all having enzyme applications for multiple sectors. So the utilization of the capacity is there for them, because invariably there are some enzymes for a specific industrial segment which they will be producing. And they are global players. So for an Indian player to set up a facility with just one enzyme for one application, I would strongly say it is going to be a challenge. It's not going to be easy. I'm sure Dr. Sham can add to this more on the practical challenge related to this. That's why I'm very particular to say, if you have a novel idea, it is something unique enzymes, work together with the global players, probably for you to be more benefited, rather than trying to think that we can probably come out with a big commercial facility without realizing the other challenges. Dr. Sham, you want to add something?
Yeah, no, I totally agree with you. We need to have a basket of enzymes to be for any company to become viable. And as you rightly said, both low-hanging fruits as well as the risky project and the challenging enzymes. So all of them have to be put in the basket for a long-term success of the company.
Correct.
And it cannot be looked at in the short term.
Yeah.
Enzyme biomanufacturing is a long-term goal. So if you are putting your foot into it, you be ready for a long journey. That's what I can say.
Yeah. I mean, it need not be a discouragement
No.
for innovation. Innovation should continue, but then specifically we suggested about the biocatalysis enzymes. These are low volume, high value products, which actually the fermenter capacity and other things could be far more lesser than a other big submerged fermentation facility for a bigger commercial requirement. So that would be our recommendation.
Thank you, sir. So this was the last question. We will take more questions if there are any offline, and we are requesting the participants to fill them, and we can take it up with them with the experts. Thank you. Over to you, ma'am.
Of this insightful webinar, I would like to extend our heartfelt thanks to all our speakers for sharing their thoughtful insights on the immense potential of the subsector on biomanufacturing of enzymes. Please mark your calendars for the next webinar in this series, which will continue to explore various thematic areas of Bio-foundry and biomanufacturing initiative. Details will be soon shared on the DBT and BIRAC websites. Please look out for the same. Thank you once again for joining us today.
Thank you. Thank you, everyone.
Thank you.
Thank you.
Thank you all.